So good evening, everyone. So just as an introduction, my name's Dr. John Hazeldine, and I'm a researcher in the Trauma and Critical Care Group based at the University of Birmingham. And I've been working in the field of trauma management and critical illness for the last 12 years. And in particular, I've focused on a study which I'm going to talk to you tonight, which is referred to as the Golden Hour Study. And we're looking to identify early biomarkers which are associated with patient outcome and ultimately try and improve the management of our trauma patients. So to set the context to our work, you'll all be aware that trauma is the leading cause of mortality in individuals aged under 44 years. And globally, 10% of deaths are attributable to traumatic injury. And the impact of this is demonstrated quite nicely on this figure here on the left-hand side, where the number of people who die each year as a consequence of traumatic injury exceeds those who have fatalities as a consequence of HIV, TB, and malaria combined. But due to medical advancements, we now know that people are surviving more serious injuries than they have ever done before. And consequently, it's a development of secondary complications that are having a big impact upon the health status of our trauma patients. And in particular, infections are the most frequent cause of poor outcome amongst our hospitalised, severely injured patients. And in terms of poor outcome, we're talking about an increased mortality risk, but also both poor physical and psychological recovery. Now, if infections are a common cause of poor outcome in our hospitalised trauma patients, this would suggest that traumatic injury has a negative impact upon the immune system, given that the immune system is our first line of defence against pathogenic infection. And indeed, if you was to Google such key terms as trauma, infections, or trauma and the immune system, you would come across a number of different papers and articles which have demonstrated how different types of injury can indeed have a suppressive effect upon the immune system. So it's well established that traumatic brain injury can have quite immediate and profound impacts upon the immune system. This has also been reported in thermally injured patients and also in patients who undergo chest trauma. Now, this was already known before we started our study here at Birmingham, but what we did is we looked at these papers in more detail, and actually we're quite interested to see that quite a lot of the information that was known with regards to how trauma impacts upon patient outcomes and the immune system was all derived from the analysis of blood samples that were taken quite a significant time after the initial injury. So this particular paper decided to analyse the immune system and bleed their patients only after they were admitted to the intensive care unit. There were other studies that only sampled their patients on days 3, 6, and 9 after trauma, where other studies took the adoption of looking at patients within the first 72 hours. So by looking at this data, we were quite interested in thinking, well, how early do these changes in the immune system happen? And if they happen very early on, can they be used as indicators of what trajectory the patient may be on when they arrive into hospital? So if we look at this in a different way, we can look at the flowchart of the patient's journey following their traumatic injury. So if we focus on those previous papers that I showed just on the earlier slide, the majority of studies were focusing on bleeding their patients after hospital admission. So the first line of in-hospital care is that some studies acquired blood samples within the accident and emergency department, where other studies actually waited days until the patient was admitted to ICU after surgery before they took their first blood sample. But as we're all aware, the patient journey following a traumatic injury actually takes place in the pre-hospital setting, i.e. at the scene of the injury. But when we first started this project, there was no study that had examined what happens in this particular time frame. So if we assume, for example, that some studies were taking their first blood sample at the time of accident and emergency admission, this could be anywhere from 4 to 12 hours after the initial injury. But what was happening at this initial stage? So to try and address this question, we came up with a study which we entitled the Golden Hour Study, and this was made possible because we established a collaboration between frontline paramedics and researchers here at the University of Birmingham. So we collaborated with the Merit team and also the West Midlands Air Ambulance Service, and once a patient was admitted to the hospital, our study was supported by a 24/7 trauma research team who were responsible for acquiring additional in-hospital research blood samples, consenting patients, and clinically following them up during their hospital stay. Currently, we are the only UK-based centre that has the infrastructure and capacity to acquire blood samples from trauma patients in the pre-hospital setting, And we have termed this the ultra early response to major trauma. So what do I actually mean by the ultra early response to injury? Well, what we've been able to do through this collaboration with paramedics is actually take our research to the scene of the injury. So rather than waiting for the patient to come to us, i.e. in accident and emergency to bleed them, through our interaction with frontline paramedics, we can bleed our patients at the roadside or at the scene of injury. And these are just some examples of the scenes that we've been able to travel to and acquire blood samples from major trauma patients. Now, currently, we've been able to recruit more than 200 patients into our study. And if we look at the average time from 999 call to the acquisition of our blood samples, this is 41 minutes. So we're really getting ultra early samples, which enables us to investigate how quickly things can happen to the body before the patient even arrives to the hospital. And our fastest sample so far has been 13 minutes after a major traumatic injury. So what have we decided to investigate in the Golden Hour study, and why have we decided to do this? Well, the two things that I will present today is the main focus is looking at the immune response and the inflammatory response to major injury. And we're trying to address such questions as what happens to the immune status of the patient within minutes of major injury, and is it feasible or possible that the health trajectory of our patients is already established during this ultra early response, i.e. in the pre-hospital setting, before the patient has even received surgical interactions or admission to A&E. And as well as the immune response, we're also interested in looking at the coagulation response. So what happens to the clotting system within minutes of a major trauma? And if changes occur in the coagulation system, is this associated again with poor clinical outcomes? So first of all, just to show you some data that we've generated with regards to the immune and inflammatory response to major trauma, this figure here, which I've put on the left-hand side, was the classical model of what was believed to happen after a major traumatic injury with regards to the immune response. So shown here was the initial insult. So this could be, say, a road traffic accident or a fall from a height. And it was believed that this initial response resulted in a robust pro-inflammatory response. So the body became activated as a consequence of the traumatic injury. And this was associated with the production of pro-inflammatory cytokines and activation of the immune system. This resulted in systemic inflammation and made the patient particularly susceptible to the development of early multiple organ dysfunction syndrome. This procedure would happen immediately following the traumatic injury and would persist for around about 1 to 2 days. Following this initial inflammatory response, this model proposed that we then had an anti-inflammatory response or a suppressive response in order to try and repair and restore the immune function of the patient. However, if this anti-inflammatory response became too powerful, it was feasible that these patients were then susceptible to the development of late multiple organ dysfunction syndrome. So we have what we call a SIRS response, so a pro-inflammatory response, and this was followed in the days to weeks by an anti-inflammatory response with the intention of restoring the patient back to their normal healthy state, which they were present before their initial injury. But with research, and this one particular study that was published in the United States, they actually questioned whether this model was actually correct. Now, in this particular study, they used both data from burns patients and major trauma patients and looked at changes in the expression of molecules that were present in the immune cells. And rather than this model, where they suggested you have a pro-inflammatory response first and then an anti-inflammatory response, this particular paper suggested that both of these responses actually happen simultaneously. So you get both activation of the immune system and also features of suppression of the immune system simultaneously. And it's whichever one of these was the most powerful determined which trajectory the patient was on. So for example, if we look here, if the patient had a high inflammatory response and this was prolonged and did not come down back to baseline, these patients were susceptible to a complicated outcome, which could have been mortality or the development of multiple organ failure. Similarly, if the patient had too much of a suppressive response, so their suppressive immune system persisted or was of an extreme case, then again, these individuals were susceptible to poor outcomes, which was related more to the development of hospital-acquired infections. Now, this study was just published before we started our work here in Birmingham, but importantly, what this study didn't do was actually look at functional responses of the cells. So they isolated immune cells from these patients, but they just looked at the expression of molecules. We didn't know from their research how these immune cells responded if they were challenged with a pathogen in the case of hospital-acquired infections, for example. So given that we had this unique collaboration with our paramedics, which we could take really early samples, the first study that would ever be able to do this, and we found that there was missing evidence with regards to how does the immune system actually functionally respond when challenged with a bacteria or a virus following an infection, we decided to create a study where we investigated the ultra early impact of major traumatic injury on the function of the immune system. And in particular, we wanted to see whether we could provide evidence that the immune system of a major trauma patient is changed or altered within minutes of injury and even before they arrive to hospital. So in this particular study, we recruit any major trauma patient that can either have a head injury, isolated head injury, an isolated extracranial injury, or patients who have suffered both head and ECI injury simultaneously. And when I say the immune system, what kind of cells am I talking about? Well, for today's talk, I'm only going to focus on two particular cell types that we have an interest within our research group, and these are called our neutrophils and our monocytes. So to provide just a little bit of background information on these cells, neutrophils are the most abundant white blood cell in your circulation, and they form approximately 70% of all leukocytes within the bloodstream. They are critical for providing protection against infections with rapidly dividing bacteria, fungi, and yeast, and they do this because they're equipped with a multitude of different antimicrobial strategies, and I'll show you some of these on the upcoming slides. So as well as neutrophils, we're also interested in a group of cells which are called monocytes. So there are a lot less of these in your circulation. Only around about 5 to 10% of your circulating immune system is made up of these cells, but they're particularly good at recognizing bacteria and engulfing or eating the bacteria. Importantly, monocytes are also a rich source of cytokines. So by this, I mean that when a monocyte responds to a bacteria or a virus, it can become activated and release molecules which will alert the body to the presence of an infection and activate other immune cells. And this particular function is something that we've looked at in our study to date. So the first question we wanted to ask is, if we're the first people to obtain blood samples so quickly after a traumatic injury, is there actually any evidence that the immune system can indeed be activated within minutes of a major trauma? So on the left-hand side here, we have our neutrophil, so our first line of defense against infection. And when neutrophils do become activated, they increase the expression of certain receptors or molecules on their surface. So the first thing that we did was bled a group of aged and sex-matched healthy controls, and we looked at the expression of a particular marker on the surface of the neutrophil, which is referred to as CD11b. We then performed the same experiment, but looking at the blood samples which were taken from our patients in the pre-hospital setting, so within 60 minutes of the injury, and what we saw is quite a significant and sharp increase in the expression of these molecules on the circulating neutrophils present within our trauma patients. So this was some initial evidence to say, yes, within minutes of traumatic injury, the cells of your immune system are being activated. And as well as neutrophils, we saw we investigated whether this was also happening in the second immune cells that we focus on here, which was the monocyte. So in this particular experiment, we looked again at a particular marker on the surface of the monocyte called toll-like receptor 2. And as you can see in this figure, trauma patients bled within 1 hour of injury were showing increased expression of this molecule on their surface when compared to healthy controls. So two of the most important cells of your innate immune system, we had evidence were being activated within 60 minutes of an injury. And as well as this activation at the cellular level, we also took blood samples and isolated serum from our patients, and we looked at certain molecules within their circulation, which are again indicative of activation of the immune system. So here we've just got 3 graphs looking at 3 different pro-inflammatory molecules. So these are molecules which will be generated in the SIRS response, so the systemic activation of your immune system. And if you have higher levels of these, it points towards increased inflammation. And as you can see, for all 3 of these markers, the patients bled within 60 minutes of their injury had higher levels of these molecules within their circulation, providing further evidence of this initial hyperactivation of your immune system. But what about suppression? So we mentioned earlier in that model where we have a SIRS response and the CARS response, well, we've showed evidence already that we have immune activation within minutes of injury. But what about immune suppression? So neutrophils are shown here on the left-hand side, and they have an antimicrobial mechanism, which is referred to as the generation of extracellular traps. So what you can see in this figure here is we have a neutrophil shown here in yellow, and it's been challenged with bacteria, which are shown in blue. When the neutrophil recognizes the bacteria and becomes activated, it releases its DNA into the extracellular environment with the intention of trapping or capturing the bacteria. This then stops the bacteria from spreading throughout the body, and it enables other immune cells to travel to the site of bacterial challenge and help clear the infection. So these are very important molecules and structures which help you protect against infection. So what we did is that we isolated, again, blood cells from healthy controls, shown here in HC, or our trauma patients at 3 different time points post-injury. So we had our pre-hospital 1-hour sample, and then, as mentioned before, when the patients are admitted to hospital, we also bleed them at a number of in-hospital time points. This is 4 to 12 hours post-injury and also 48 to 72 hours post-injury. And hopefully what you can see here is that when we challenged healthy control neutrophils with bacteria, we got an increase in the generation of these extracellular traps, so they can help capture and remove the bacteria. But hopefully what you can see is that within 1 hour of injury, when we challenge our patient neutrophils with a bacteria, their production of these extracellular traps was reduced. And this persisted to both 4 to 12 and 48 to 72 hours. So we've got some evidence here of immediate and also persistent immune suppression following major trauma. And as well as neutrophils, we looked at how monocytes respond to bacterial challenge. So in a healthy individual, when we're exposed to a bacteria, your monocytes will become activated and they release into the environment these inflammatory cytokines, which signal to the body and to alert other immune cells that we have an infection and we need to increase our immune activity. So what we do in the lab is that we bleed either healthy controls or our trauma patients, and we expose them to bacteria to try and mimic this infection, which could happen while they're hospitalized. So in this particular experiment, we took E. coli and we challenged blood samples for 4 or 18 hours with the bacteria. And then after these particular time points, we studied how active the immune system was. And hopefully, again, what you'll be able to see here is we looked at 3 markers which are produced by activated monocytes. And compared to our healthy controls, you can see that in response to bacterial stimulation, the monocytes from our pre-hospital patients was significantly reduced. So already within 1 hour of the injury, the ability of your monocytes to respond to bacteria is significantly impaired. And as well as function, I also mentioned earlier that in response to a hyperactivation of the immune system, the body responds by undergoing a CARS response or an anti-inflammatory response. And again, what we decided to do was measure the levels of anti-inflammatory or immune suppressive molecules in the serum samples of our patients. And what you can see both for IL-10 here and also IL-1, that our trauma patients were presenting within 60 minutes with increased levels of these markers in their bloodstreams. So again, we're collecting evidence to show that within 1 hour of injury, before your patient arrives to hospital, is showing signs of immediate immune activation, but also suppression. And it will be the balance between these two which determines the health status of the patient. So if we kind of know what happens to the immune system after a traumatic injury and how early this occurs, we were next interested to see what factors can contribute to this immediate immune suppression. Because if we know that immune suppression happens, if we can identify the factors or the molecules which drive this suppression, then it's identifying by improving our understanding some potential therapeutic targets that could be used to develop novel treatments. So one of the first things we thought about was, well, how are immune cells produced after a major trauma? So we know that neutrophils are produced in your bone marrow, and they undergo a number of sequential steps from an immature cell within the bone marrow to your mature neutrophils, which are released into your bloodstream and can function to protect you against bacteria, fungi, and yeast. So from the left-hand side to the right-hand side of this screen, the maturity status of your neutrophils increases. So in the bone marrow, you have the most immature forms of your neutrophil, and they progress through these transitional steps until they form what's called a mature neutrophil, which is then released into the circulation. Now, this first part of neutrophil development, shown by these first 5 circles here, are collectively referred to as immature neutrophils or immature granulocytes. So we thought maybe trauma resulted in increased number of these immune cells within the circulation. So with blood samples that we took from our patients within 1 hour of injury, 4 to 12 and 48 to 72 hours, we ran them through a whole blood analyzer, which we have in the lab, and we looked specifically for the number of these immature cells. And hopefully what you can see here is that we have a high proportion of these cells within our blood samples, which were taken from patients within 1 hour of injury, and then this declined over time, but their levels still remain significantly higher than what we observed in patients who were healthy controls. So, as I've mentioned previously, one of the key functions of neutrophils, so how they help us protect against bacteria, fungi, or yeast, is a process which is termed phagocytosis. So hopefully what you can see in this image here is we've got our activated neutrophil, and it's recognized a bacteria. In response to becoming activated, the bacteria is captured, engulfed, and then destroyed. Now, interestingly, if you look at the ability of immature and mature neutrophils to perform this function, we actually observed there were quite significant differences in their ability to do this. So shown here in this figure is our immature cells and our mature neutrophils. So what we did is we looked at how active these 2 different types of neutrophils were in the bloodstreams of our patients. So this is a trauma patient, and when we looked at the mature neutrophils in the trauma patient and how well they could eat bacteria, we could see that 97% of these mature neutrophils were able to recognize and eat bacteria. But when we looked at the immature neutrophils, hopefully what you'll be able to see here is that there's a significantly reduced ability of immature neutrophils to recognize and eat bacteria. So if our trauma patients have higher levels of these immature cells within their circulation, this may well be contributing to the impaired immune system, which can happen within minutes of traumatic injury. Now, a common feature of traumatic injury is obviously significant tissue damage. So we were interested in thinking about whether molecules are released from damaged tissue that can then suppress the immune system. So this figure here just shows you the different types of cellular damage that can occur following a major trauma and the kind of molecules that this can result in being released into the circulation. Now, we had a particular interest in one molecule, which was called N-formulated peptides. So these molecules are normally found inside mitochondria, so inside organelles within the cells, and you do not find them normally in the circulation of a healthy individual. However, in times of tissue damage or cellular stress, these molecules can be released into the circulation. So we were interested to see how quickly these molecules are released. So we took serum samples from our trauma patients and we looked for the levels of some of these molecules which are released from damaged tissue. And again, what we were able to see was that within 1 hour of injury, we can pick up high levels of these molecules released from damaged cells and damaged tissue, showing that we have an immediate release of factors into the circulation. And importantly, these factors persist for up to at least 3 days following the traumatic injury. So we have an immediate release, which then persists above the levels which are observed in healthy controls. So if we have high levels of these molecules, does it actually mean anything? Can they indeed promote immune suppression? Was our hypothesis along the right lines? So another molecule which is released rather than from cells but from damaged muscle tissue is a molecule called heme. So within our muscle we have myoglobin, and myoglobin contains a particular molecule which is called heme, and this is responsible for binding oxygen. So we would have thought if we have tissue damage, we have muscle damage, is this molecule heme also released into the circulation from the myoglobin which is stored in the muscle? And indeed, what we were able to show, again, within 1 hour of injury, is that you see high levels of this molecule heme within the circulation of our trauma patients. But this seems to be a really early marker of damage because its levels reduce over time to the point of by 48 to 72 hours, the levels are similar to those that are observed in healthy control. So this is really an early immediate marker of tissue damage. So if we know that molecules are released from damaged tissue into the environment and into the bloodstream, Can we mimic the immune suppressive effects that we observed in a trauma patient by treating healthy immune cells with factors which are released from damaged tissue? So in these experiments, what we decided to do was get a group of healthy volunteers, bleed them and isolate immune cells from their blood samples and expose them to molecules which are released from damaged tissue to try and mimic a traumatic injury in the lab. So this is the figure that I just showed you a couple of slides ago, where I was able to show that within 1 hour, 4 to 12, and 48 to 72 hours after a traumatic injury, neutrophils show reduced ability to generate these neutrophil extracellular traps, so reduced response to bacterial stimulation. Now, interestingly, what you can see here is that when we took neutrophils from healthy donors and then exposed them to these molecules which are released from the damaged tissue, we could mimic the effects that we were observing in our actual trauma patient samples. So on the left-hand side, these are healthy control neutrophils which have not been exposed to any damaged molecules. They generate lots of these extracellular traps. But when you treat them with molecules released from damaged muscle or damaged tissue, they have an ability or an inability to respond to the bacterial stimulation, which suggests that circulating factors could be contributing to the reduced immune response of our patients. As well as neutrophils, we also looked at monocytes. So again, this figure, if you remember, is what happens when a monocyte recognizes a bacterial stimulation. It produces these inflammatory molecules. I showed you earlier that within 1 hour of injury, the ability of a trauma patient's monocytes to release these molecules is reduced. And this reduction happens at the same time as which we get the release of heme from our damaged muscle. So our myoglobin is being released into the circulation. So what we did in these experiments is take healthy control monocytes and expose them to concentrations of heme, which you detect in a trauma patient, and then expose them to bacteria. And when we did this experiment, what you can see here is that when you pre-treat healthy control monocytes with the molecule from damaged muscle, this reduces their ability to respond to bacterial stimulation. But importantly, we know that there are molecules which are in the circulation which can bind to this molecule heme and reduce its levels in the circulation. And these two molecules are called hemopexin or albumin. So these are proteins which we find in our plasma. And if these proteins are present within your plasma, they can bind to heme and prevent it from being immune suppressive. So this is a potential therapeutic option that we have, that if we know that heme levels are high after trauma and heme can reduce immune function, then could we mop up or clear any existing heme by treating patients with hemopexin or albumin? And when we measured levels of albumin and hemopexin in the blood, we were able to show that on day 1 of injury and also day 4 to 12 hours and 48 to 72 hours, that the levels of these molecules, both albumin and hemopexin, are significantly reduced when compared to healthy control. So our trauma patients not only have high levels of damaged tissue and released myoglobin into the circulation, they also have reduced levels of the molecules which can help prevent the immune suppressive effects of heme. So we kind of have a vicious cycle here of tissue damage and also reduced molecules that can prevent immune suppression. But importantly, we know that these are feasible targets for treatment patients and could offer a potential therapeutic avenue by which to try and reduce the level of immune suppression that happens following major trauma. So this raises the question of future studies which we could work on is, could restoring the levels of these two molecules in the bloodstream of trauma patients help reduce trauma-induced immune suppression? So just moving slightly away from immune suppression, I thought I'd just show you a few slides where we're looking at unique insights which are discovered by the analysis of pre-hospital blood samples. So like I've said before, we're the only UK centre that could gain access to these blood samples. So we were interested in seeing if there is anything unique within these blood samples that has not been previously reported in patient samples that have been taken after hospital admission. So again, referring back here to neutrophils and the generation of these extracellular traps. One particular study wanted to see whether these neutrophil extracellular traps are generated following trauma where there is evidence of no infection. So this is just a sterile traumatic injury, and this group looked in 7 patients to see whether they saw any evidence of this activation of the immune system. And compared to patients who did have an infection, they reported that there was no evidence of these molecules being generated following a traumatic injury. But when we looked into their data, they actually said, or when they concluded their work, that they believed that the timing of their blood samples was too early to detect the evidence of these nets after the onset of critical illness. And when we looked at when they took their blood samples, they waited until their blood samples were acquired within the ITU. So the patient had to be admitted to the ITU ward before they were sampled for this immune analysis. So they suggested that they needed to wait even longer in order to detect these molecules in the circulation. What we hypothesized is, could they be incorrect in is that rather than being too early, they were in fact too late and actually missed this particular component of the immune response to traumatic injury. So what we did is we took our plasma samples from our traumatically injured patients and we screened for markers of these extracellular traps in the circulation. And hopefully what you can see here is that in these 3 different trauma patients, we were able to detect in our pre-hospital samples evidence of these extracellular traps being generated in response to the traumatic injury. But interestingly, when we followed the same patient across multiple time points during their in-hospital stay, you can see that we were unable to detect this marker in samples which were taken 4 to 12 or 48 to 72 hours after the injury. And it was only in these pre-hospital samples where we were able to see evidence of this immediate immune activation. So rather than being too early, the actual previous study was too late in terms of they waited too long in order to observe this response of the immune system. And again, this is just to show this visually. You can see that in samples taken with 1 hour of injury, you can see the generation of these structures or these extracellular traps, but these structures have disappeared by 48 to 72 hours after the injury. And one other unique insight, which I thought I'd just share, is looking at different types of immune cells within the circulation. So again, at the time that we started this study, it was previously thought within the literature and a well-accepted phenomenon of major trauma that the number of circulating lymphocytes that a trauma patient has in their circulation is significantly reduced when compared to healthy individuals. And indeed, if you look at this graph, we bled healthy controls, and when we acquired blood samples from our trauma patients at 4 to 12 or 48 to 72 hours after their injury, their levels were indeed significantly lower than that was observed in healthy controls. But interestingly, in our pre-hospital or within 1 hour blood sample, you can actually see the number of these immune cells are actually increased in trauma patients rather than being reduced. So again, this was providing new insights into how the immune system and how the body responds to a major trauma within minutes of the injury occurring. So how do these ultra early changes then associate or relate to the outcome of the trauma patient? So we're particularly interested in looking at the development of multiple organ dysfunction syndrome amongst our critically ill patients. And multiple organ dysfunction syndrome has been reported to have an incidence as high as 56% in adult ICU patients. And it's the most common cause of late trauma deaths, with studies estimating anywhere between 20 and 75% mortality in patients who have at least failure in 2 separate organs. So if I take you back to this graph here, where I showed you that within 1 hour of injury, the number of lymphocytes, so a type of immune cell within the bloodstream, was significantly higher in 1 hour of injury when compared to healthy controls. And this had not been described before. So there are many different types of lymphocytes within your circulation, so many different subsets or classes. And one particular subset is referred to as an NKT cell. So what we decided to do was count the number of these cells in the circulations of our trauma patients when we split them into those who did or did not develop multiple organ dysfunction during their hospital stay. And hopefully what you can see from this figure here is that within 1 hour of injury, so in the pre-hospital setting, those patients who had higher levels of this particular type of lymphocyte within their circulation, so the NKT cell, went on to develop MODS when compared to those patients who did not. So you have an early distinguishment within 1 hour of injury by looking at one type of immune cell, which identifies or suggests that particular subtypes of patients are susceptible to the development of multiple organ dysfunction. And when we looked at data at 4 to 12 and 48 to 72 hours, this relationship was not observed. So we could only see this difference in these 2 patient groups by using data which we had acquired in the pre-hospital setting. And just to put this into context, What you can see is you could imagine you have two patients who arrive in the accident and emergency department with the same base deficit and the same injury severity score, but one patient had 84 million NKT cells in their circulation and the other patient had 185 million cells in their circulation. So what our analysis suggested in this small cohort study was that the patient who arrived in hospital with the higher number of NKT cells had a greater than 60% chance of developing multiple organ dysfunction syndrome, even though they had the same injury severity score and the same baseline deficit at the time of hospital admission. So this is suggesting that acquiring or the acquisition of pre-hospital blood samples and looking for certain types of immune cells could help potentially identify patients at risk of poor clinical outcomes. And another marker which shows promise as a potential indicator of poor outcomes is cell-free DNA. So when cells get damaged and tissue gets damaged, DNA can be released into the circulation. And we were able to show that in our study that again, within 1 hour of injury, the amount of DNA that you could detect in the circulation of our trauma patients was significantly greater than that was observed in healthy control. And the levels were higher in the 1 hour sample when compared to your in-hospital sample time points. And again, what we did here is we divided our patients into those who did or did not develop multiple organ dysfunction. And we were able to see that within 1 hour of injury, those patients who presented with higher levels of cell-free DNA after their injury went on to develop multiple organ dysfunction syndrome compared to those who had better outcomes, i.e. no multiple organ dysfunction syndrome. So just moving slightly away from the immune response and to finish with some data with regards to the bleeding response or the coagulation system, does this also change within minutes of injury? So in terms of the coagulation response, it's well known that after major trauma, you get disruption to endothelial cells, and this can impact upon the ability of the blood to clot and also have an impact upon your immune system. But no investigations had actually looked at this process in the pre-hospital setting. In other studies that had looked at endotheliopathy or damage to endothelial cells, they looked at two particular markers. One was called thrombomodulin and one was called syndican. If you have high levels of these markers in your circulation, it's indicative or used as a marker of disruption of the endothelial barrier. But it was unknown exactly what time or how quickly this occurs following major trauma. So what we did is measure the levels of these two molecules in our within 1 hour blood samples, and we were able to show evidence of increased markers in our trauma patients when compared to healthy controls. So within minutes of injury, we're showing signs of endothelial damage or disruption. But exactly how quickly did this occur? So what we did in this particular study was measure the levels of these markers in healthy controls, which is shown by this blue line here, and then in our trauma patients, which are shown in a red line here. And hopefully what you'll be able to see by this dotted line is that we use mathematical modeling to predict exactly how quickly your endothelial cells are damaged or dysfunctional following trauma. And our data suggests that within 5 minutes of a traumatic injury happening, your endothelial function is already significantly reduced, which could have impacts upon the clotting response, but also your immune system. And again, this had not been reported previously because all other studies had waited until hospital admission to bleed their patients. So endotheliopathy or dysfunction of the endothelial system is what we call an on-scene phenomenon. And is this related to any degree of patient outcome? Does it have an impact upon how patients develop secondary complications, for example? So again, focusing on multiple organ dysfunction, hopefully what you'll be able to see from this figure here is that high levels of syndican-1 and also thrombomodulin were detected in the pre-hospital time points of those patients who went on to develop multiple organ dysfunction syndrome compared to those patients who didn't. So again, we have a particular evidence or some evidence to suggest that early sampling of pre-hospital patients might have the ability to identify markers which could help predict patients who might be at risk of poor clinical outcomes. And then finally, just rather than discussing about what happens to the immune system, we were also interested in seeing whether we could look for biomarkers that help diagnose the severity of a particular injury. So here we focused on patients that had traumatic brain injury and had an idea that we need to develop reliable biomarkers which could help improve the management of our patients and potentially result in a development of new clinical interventions. So what we did in this particular study was take serum samples, again from our traumatically brain injured patients, with or without extracranial injury. So this axis here shows healthy volunteers shown by HV, patients who just had an extracranial trauma, patients who had a mild traumatic brain injury with extracranial injury, or patients who had a severe traumatic brain injury with extracranial injury. And what we were able to see is we identified this particular marker in the circulation of the patients called CTS5, and we were able to show that this marker was increased in patients who had a severe traumatic brain injury when compared to those who had a mild traumatic brain injury. And these differences were most evident in the less than 1 hour blood sample when compared to the samples which were taken 4 to 12 hours after the injury. So we have a potential marker here which helps delineate between a patient who has a severe traumatic brain injury compared to a mild traumatic brain injury. So we believe that this is worthy of further investigations in order to identify or the context of an ability to detect severe traumatic brain injury within the pre-hospital setting. And then finally, as well as looking at that particular marker, we also looked at what were called microRNAs to help in the diagnosis of mild and severe traumatic brain injury. So in this particular study, we analysed samples taken from 120 of our trauma patients at our 3 sampling time points and looked for the presence of what are known as microRNAs. So these are molecules which are released from cells, either activated cells or damaged cells. And what we were able to do is we found 2 particular types of these molecules which were significantly downregulated in patients who had suffered a mild traumatic brain injury. And conversely, we found 2 types of these molecules which were significantly increased in patients who had a severe traumatic brain injury. And trying to relate this to outcomes, so do the levels of these markers mean anything in terms of patient trajectories or patient medical outcomes, we found that one particular type of this molecule released by these activated or damaged cells following a traumatic brain injury was a strong predictor of 6-month outcome. So again, we're trying to accumulate and generate data to try and help us find useful biomarkers to help predict patient outcomes after, in this case, a traumatic brain injury. So we've identified some potential molecules which could show promise as biomarkers to discriminate between different severities of traumatic brain injury. So just to summarize the talk I've gone through today, we've been able to establish for the first time that within minutes of major trauma, the immune system displays signs of activation and suppression. Our own data, as well as that derived from earlier studies, suggest that it's actually the robustness or the magnitude of this response, as well as how long it persists for, that can influence patient outcome. It appears that the timing of your blood sampling can impact upon how you understand how the body responds to an immune and inflammatory response. We've been able to identify unique responses in our pre-hospital blood samples, which aren't detectable in samples that are taken after the patient has been admitted to the accident and emergency department. And finally, by investigating ultra-early immune or coagulation responses, there's some data to suggest that acquiring these blood samples could be useful in identifying patients who are on what we call a negative clinical trajectory. So identifying those at risk of potential poor outcomes. So that just leaves me to thank all of the people involved in this study. So obviously you can imagine it's quite a big collaborative study involving both paramedics, research nurses, and also academics here based within the University of Birmingham. And we also like to acknowledge all of the funders who've been kind enough to support our work here today. So thank you, and I'm happy to take any questions.
Lovely stuff. Just stop sharing that, and then I can get the usual screen up, if we can find it, as per usual. Keynote, roll blood. Excellent. I think we're on the right one. We are indeed. We are indeed. Right, lovely stuff. Thank you very much, John. A fascinating talk. Someone beat me to it. Ingrid did, saying a fascinating talk. I mean, the science behind the trauma is always so important. So we are so grateful. And the talk has already very quickly brought in a lot of questions. So before I jump onto those, I just want to draw everyone's attention to our next webinar. We're going back to a topic that we haven't touched on in a while, actually. So we are extremely, extremely grateful and excited to welcome Bob Handley to Trauma Care again, because we've had Bob quite a few times before. However, he's going to be joining us on March the 6th. So that's literally 2 weeks from now and about 2 days before the conference begins. And he's going to be talking about addressing unwarranted variation in the care of older people. And this it's not a preachy sort of webinar that's going to be telling you should be, it's looking at a standardized level of care between all types and how we are taught, how healthcare professionals are taught to sort of treat people the same, you know, as James Nair from Kid Confidence always says, they're just mini adults. And this is sort of the same style of that and how to approach care of the elderly. So if you want to scan the QR code that's on your screen now, you are welcome to join us then. However, as we already have 9 questions in, I'm going to jump straight to the Q&A. The QR code that's on your screen now is for all you non-members of Trauma Care. So those of you that are joined us tonight and are not members of Trauma Care. If you scan the QR code that's on your screen now, it will take you through to the type form. Once you've filled this out, all you have to do is pay £1.99 and the certificate will be sent straight to your inbox. Don't worry, type form, it's not another registration, it's not you signing up to anything, it doesn't hold your data or anything like that. It's just for us so I can create the automation. So there you go. So we shall begin at the top. Mark Wilson has asked, are you taking blood at cannulation? How are you keeping blood viable when extrication and transport is long?
So all of our samples are, I guess, taken, well, cannulation if they have a cannula, or venipuncture if they don't. All of the samples are processed within 1 hour of their deposition within the accident emergency department. So our variability is at a minimum. In terms of how we process the blood samples, all of the blood samples are processed by the same single laboratory researcher. So there's no variation in terms of the experimental approach. And they are all stored, or delivered to us at controlled temperature conditions. So the longest we've ever had to wait between acquisition of a blood sample in the pre-hospital setting to then actually arriving here at the QE has been 1.5 hours. And we've done some studies in our own healthy control work where we've been able to show that if you bleed a healthy control and you leave it for these different periods of time, it doesn't affect their functional response. So we know that if we have a sample which is delivered within, say, 40 minutes of the accident happening, actually in our hands to do the laboratory research, compared to another one which was 90 minutes, there isn't actually any difference in that time frame between the functional responses that you can see. So we try and minimize as much variation, both from the experimental side and also the transporting side, to give some consistency to our results.
Wonderful, thank you. And again, a very thorough, fascinating answer. Tim, hello, Tim. Hello, Professor. So Professor Tim Coates has asked, really interesting parallels with Misha Heron's early pre-hospital coagulation blood sampling in the London HEMS system. Do you think that, given the number of molecules in common, interactions between coagulation and immune systems are likely to be important?
Yes. So the only thing I can say from our side in terms of the research we've actually done, we've looked at interaction between red blood cells, platelets, and white blood cells, particularly neutrophils, and how their interaction after trauma can modulate the function of the immune cell. So we've been able to find after trauma, and particularly within that 1 hour sample, we have high levels of platelets binding to neutrophils. So we have interaction between the blood system and the immune system. And those cells which have these red blood cells bound to them actually increases their functional responsiveness or their activation. So we don't have lots of data on that, but the initial data that we do have is that this interaction between the blood cells and your immune cells happens immediately. So within that 1 hour sample, the levels are the highest. And in those cells that do have the interaction between the two cell types, they're showing different responses when compared to those white blood cells that haven't had an interaction with the red blood cells.
Wonderful. Thank you very much. An anonymous attendee has asked quite simply, what's the difference between natural killer cells and NKT cells?
So basically that's just the difference in what they recognize. So natural killer cells are your first line of defense against virally infected cells or cancerous cells. And NKT cells are more involved in the recognizing of lipids. So basically like fatty acids within the bloodstream. So they use similar functions. They behave similarly when they're activated, but it's what activates them which differs between invariant natural killer T cells or iNKT cells and your NK cells.
Wonderful, thank you. Liam Higgins has asked, if I've arrived on scene before the ambulance and controlled the bleed, could I include any info in my APMIST, such as immunoresponse, to help assist that patient in further care?
I'm not a clinician, so I don't really know what to say to that. The only thing that I can kind of say from what our research is trying to do is be able to see if we can develop something in the long term which would give you an on-site understanding of the immune response. So obviously at the moment we still have to bleed the patient, wait for the samples to come into the hospital to then perform the experiments. But some of those markers that I spoke about earlier, so for example the immature cells, we're trying to develop, or people are trying to develop, ways of being able to detect this through just like say a little pinprick, and then develop the same answer as what we would do in the lab. So it's trying to basically increase, or the speed at which we could potentially generate some informative data to suggest that the paramedic is observing a patient who is already showing signs of immune suppression.
Wonderful, thank you. And Tim again has asked, could you look for an effect of TXA on immune reaction to trauma, testing hypothesis that controlling... Tim, really? Fibroneurotic cascades can, via common activation molecules, also control the immune response. The background here is trying to work out why TXA has a big effect on outcome when it is such a weak antifibrillary... I cannot say that word.
Yeah.
I apologize. I think you know what I mean.
Yeah. So the only thing I can say to that is we do have obviously the clinical data of those patients who did or didn't have TXA prior to our blood sampling, and we can try and examine whether the immune response is different between the two. So we know from other people's studies, mainly from like Australia, that yes, TXA can have a quite a significant impact upon immune function. But ourselves, we haven't really dug down into that at the moment. But we do have the database and the samples which would enable us to look at that. Like I said, we're over 200 patients now, so we have a decent-ish number to see if we could pick up a signal or a relationship between TXA, no TXA, and how this affects the immune response or the patient outcomes. But it's just something that we haven't directly tested at the moment.
Wonderful. Thank you. And the final two questions. So if anything comes in after this one, guys, I'm really sorry, but we won't be able to get around to them in time, just because this building gets locked up after a bit. So Ingrid Petrie has asked, if blood clotting ability begins to be compromised around 5 minutes after trauma, does this mean that in cases of bleeding, then stemming, that bleeding within the first 5 minutes, e.g. by bystanders, becomes even more important?
Yes, that would be our way of thinking. Yes, so the damage that we're seeing, or the markers that we use, is that rather than being a direct measure of coagulopathy or blood clotting, it's more of looking at endothelial dysfunction, and we make the hypothesis that this dysfunction, which is happening within 5 minutes, would indeed impact upon the ability of the blood to clot.
Wonderful.
So
Thank
yes,
you.
we think that the quicker you can stop that, the better. Yes, it would have a positive effect.
Wonderful. Thank you very much. I appreciate that. So stop the bleed, everybody. So, and then finally, an anonymous attendee has asked, can you please share the links to the papers on traumatic brain injury, CTS5 and MENA and TBI? Many thanks. So that's just pop that in the chat and we'll
so
go
if
from there.
you can either Google my name, because I'm an author on both of those, or Tony Belli. So Antonio Belli is the PI of that trial, and he's one of the neurosurgeons here. But I'm happy to share the links if you can't find them. Not a problem.
Lovely. Wonderful. And that is all the questions we have time for this evening. So thank you very much, John, for joining us this evening. And before we pop off and say goodbye to everybody, just another thank you to our lovely sponsors of Trauma Care, Slater and Gordon, of course, who we mentioned at the top of tonight's presentation, and QualSafe, who again you will catch at the Trauma Care conference. They've got a stand. They'll be there all week. Hopefully Toby's there all week with Nigel. They're doing various talks throughout the entire event, and they also are leading their own stream on the first day, the first aid stream. So do come and check them out. Ignore the awards bit on the website address there. Just go to qualsafe.org or scan the QR code to find out more about them. And of course, a big thank you to our long-standing partners and good friends over at Galen. I am not allowed to say too much about them because they are a pharmaceutical company and there's laws and legalities around that. However, if you would like to know the mystery behind my evasiveness there, do scan the QR code that's on your screen now. Click on I'm a healthcare professional and find out more about everything they have available. Again, they will be at the conference all week, and you all know them by now, so come and say hello. And that is everything from us this evening. So, Dr. John, thank you so much for joining us for such a fascinating talk and a very something that I I believe personally, this is me that's not clinical at all or trained in this area. I think it's something that could potentially shape the future of trauma care, especially in pre-hospital management. So thank you for sharing a glimpse into the potential future and for taking time to be with us this evening. And to everybody else, no matter where you are in the world, I've seen people tonight from Kenya, Baghdad, from all sorts of wonderful places, and America, of course, our lovely friends over at TCAR. Thank you very much for being with us this evening. Wherever you are in the world, take care of yourself, take care of your loved ones, and enjoy the rest of your night. Good night, all.